So we've talked our way through all of cellular respiration. Start off in glycolysis, then if there's oxygen present, we go to the citric acid cycle by first converting pyruvate into acetyl-CoA during pyruvate oxidation. Then from the citric acid cycle, we bring all of our NADH and FADH2 to the electron transport chain, where we make a lot more ATP via chemiosmosis from our oxidative phosphorylation instead of our substrate level phosphorylation. So what happens if there's no, that's not the beginning. What happens if there's no oxygen present? Glycolysis is still going to happen. Glycolysis always happens as the first step to break down glucose and to get energy from glucose. We split our glucose in half. During glycolysis, we get 2 ATP overall via substrate level phosphorylation. We get 2 NADH, something like that. We get NADH and we get pyruvate molecules. If there's oxygen present, that pyruvate goes into the mitochondria, gets oxidized, etc. If there's not oxygen present, there's 2 things that can happen. Basically, the electron transport chain is not going to work because there's no oxygen to accept the electrons at the end, and the whole thing's just going to back up and no ATP is going to be made from the oxidative phosphorylation without any oxygen. So then after this, one of 2 things can happen. We can either go into anaerobic respiration, which still has an electron transport chain. So anaerobic still uses an electron transport chain, except rather than having oxygen accept the electrons at the end of the chain, there's another electronegative molecule. So remember that oxygen is the most electronegative thing in the electron transport chain, and that's why it's at the bottom, because it pulls the electrons towards it and accepts them at the end. So if we use a different electronegative atom, such as sulfur, then that can also pull the electrons down and accept it. So there are some organisms, some bacteria that use sulfur as the final electron acceptor and still use an electron transport chain, except they have no oxygen. They use sulfur instead, and that's anaerobic respiration. So anaerobic respiration still has an electron transport chain. This is different than fermentation, which does not have an electron transport chain. In fermentation, the only ATP is made from glycolysis. So remember, like I just said, glycolysis gives us 2 ATP overall. 4 are made, but 2 are used, so overall 2. It gives us 2 NADH. So what happens is it just does glycolysis over and over and over again. But the problem is that we suddenly have all of these NADH molecules, and we don't have an electron transport chain to send them to. So now we have way too many NADH molecules and not enough NAD plus to accept electrons during glycolysis. So the fermentation is the process of glycolysis happening over and over again, making ATP via substrate level phosphorylation instead of an electron transport chain. So there are 2 types of fermentation, and the way that these differ is in the way that they recycle NADH. So the problem with fermentation is that we are just building up our NAD plus or our NADH levels, and we don't have enough NAD plus because the 2 are recycled. So what we have to do is find a way to turn NADH back into NAD plus, which can then be used again by glycolysis to make 2 ATP via substrate level phosphorylation. So there's 2 kinds of fermentation. There's 2 kinds of fermentation: alcoholic fermentation and lactic acid fermentation. So these 2 things differ in the way that they regenerate NADH. So in alcoholic fermentation, what happens is pyruvate is converted into ethanol, which is an alcohol, so that's why it's called alcoholic fermentation, and a CO2 molecule is released. And when this happens, we recycle our NADH back to NAD+. So alcoholic fermentation uses 2 steps. This here is alcoholic fermentation to recycle NADH. So during glycolysis, we make 2 ATP and we make 2 NADH from 2 NAD+, and we get a pyruvate out. So that's all well and good, and this could keep happening over and over and over again because we're going to have a big supply of glucose, but we need to turn this NADH back into NAD+. So what happens is pyruvate is converted into this molecule called acetaldehyde, and carbon is lost there in the form of carbon dioxide. And then acetaldehyde is reduced by NADH, so NADH gives its electrons to acetaldehyde. NADH becomes NAD+. It was oxidized. It gave away electrons, so it became oxidized. And now it can go back into glycolysis and happen over and over again. And acetaldehyde is reduced to ethanol, which is an alcohol, and it's the alcohol that you drink if you are consuming alcohol. So this process is actually how alcohol is made, by alcoholic fermentation. And then this is released as a waste product. So alcohol is actually a waste made by yeast, usually. So in alcoholic fermentation, glycolysis happens. We get to ATP, we get to NADH, and we get to pyruvate. Then the ATP leaves to perform cellular work. The NADH needs to be recycled. So what happens is pyruvate is converted into 2-acetaldehyde. We lose a carbon dioxide, 2 because there's 2 pyruvates, but we lose 1 carbon dioxide for every pyruvate. Then this acetaldehyde gets reduced by NADH. It takes electrons from NADH, becomes 2-ethanol, which is released as a waste product, and the 2 NAD+ are recycled to go back to glycolysis. So that's alcoholic fermentation. Then we have lactic acid fermentation, which is a little bit different. The goal is still to recycle the NADH back to NAD+. But what happens is we have a different byproduct. Instead of alcohol, we have something called lactic acid. And what happens here is pyruvate, rather than being converted into something else, pyruvate directly gets reduced by NADH. So pyruvate is right here. It's a byproduct of, or it's a product of glycolysis, same as over here. So we have glucose being broken in half into pyruvate. 2 ATP are made, 2 NADH are made. But now pyruvate comes and is reduced by NADH. So NADH gets oxidized, gives up its electrons to pyruvate, and it is recycled to NAD+ and can go back into glycolysis. This pyruvate, after it's reduced, becomes lactate, which is a waste product, and is released as a waste product. And so that's why it's called lactic acid fermentation. So these two are very similar. The whole goal is to recycle NAD+ so that glycolysis can continue. Because if we didn't do that, NAD+ would become unavailable as NADH builds up, and then glycolysis couldn't happen. And in these organisms, their main source of ATP is this 2 ATP made in glycolysis right here. The only difference is that in lactic acid fermentation, pyruvate directly gets reduced by NADH, whereas in alcoholic fermentation, it is converted into acetaldehyde, which then is reduced by NADH. So lactic acid fermentation is used by some fungi and bacteria, and can be used to make cheese and yogurt. And our cells do fermentation during strenuous exercise. So our cells need ATP. If you're like sprinting or lifting weights, need a lot of ATP to move those, to actually move your muscles. And so if aerobic respiration, so glycolysis, citric acid cycle, and electron transport chain are not providing enough energy, your cell is also going to turn on the pathway of lactic acid fermentation. What that means is it's going to start breaking down some of your glucose into, or it's going to start recycling some pyruvate into lactic acid in order to help more glycolysis happen. So lactic acid is released as a byproduct, and lactic acid builds up inside of your cells and is then taken away to your liver to be recycled or to be broken down. So our cells perform lactic acid when oxygen is low. And so comparing the two, comparing fermentation with anaerobic and aerobic respiration. So there's 3 pathways. They all contain glycolysis. Glycolysis happens in all of them. In the main one we studied, glycolysis comes first, then pyruvate oxidation, then citric acid cycle, then oxidative phosphorylation through the electron transport chain. That is aerobic respiration. Then we have anaerobic respiration, which is the same thing as before, except oxygen is not the final electron acceptor. This time, something else, a different electronegative atom is going to be the final electron acceptor. So we still have glycolysis, we still have the citric acid cycle, and we still have the electron transport chain to accept the oxygen, or to accept the electrons at the end. We just, instead of oxygen, have a different molecule. Then we have fermentation. Fermentation is simply glycolysis and an extra step, so glycolysis can continue to happen over and over again. In all three, NAG+ is the oxidizing agent that accepts the electrons during glycolysis. But all three of the processes have different final electron acceptors. In fermentation, it's an organic molecule, such as pyruvate or acetaldehyde. Pyruvate in lactic acid fermentation, acetaldehyde in alcoholic fermentation. These guys accept the electrons from NADH, whereas in cellular respiration, it's oxygen that it finally accepts the electrons, or sulfur in anaerobic respiration. So I know my picture's over this, but in cellular respiration, we get out about 32 ATP, 30 or 32 ATP per glucose molecule, whereas in fermentation, we only get 2 ATP. So that's why our cells mostly do cellular respiration, because it's so much more efficient. We're getting so much more energy to perform cellular work from one glucose molecule. Then just a couple of diversity of life things. Some organisms are called obligate anaerobes, so they're obligated to be anaerobic, which means that they perform fermentation or anaerobic respiration, one of the two without oxygen, and they can't live in the presence of oxygen, so they'll die as soon as they come into contact with oxygen. Then there are some things called facultative anaerobes, which means that they can do fermentation or cellular respiration. So they can live with or without oxygen. This sentence: in a facultative anaerobe, pyruvate is a fork in the metabolic road that leads to two different catabolic routes. So what that means is pyruvate is always made. Glycolysis happens and pyruvate is made from glucose. Now, when it gets to pyruvate, this is when decisions are made by the cell. Either that cell can use oxygen if it's present and continue down the cellular respiration route, or it can, if there's no oxygen present, it can recycle the pyruvate, recycle the NADH, and continue performing glycolysis through fermentation. Two alternative catabolic routes, meaning we're breaking down something. Catabolism is breaking things down. We can either do cellular respiration or fermentation, depending on whether or not oxygen is present. So here it is again. Glucose enters the cell, and glycolysis happens every single time. Glycolysis happens whether or not oxygen is present, no matter what kind of living thing it is. We get pyruvate. Then, if no oxygen is present, fermentation will happen, meaning we'll get ethanol, lactate as a byproduct as we recycle our NADH. Or if oxygen is present, it'll go through aerobic cellular respiration. We'll change pyruvate into acetyl-CoA, the citric acid cycle will happen, and then oxidative phosphorylation will happen in the cristae of the mitochondria. And that's all.